Today's lesson is a continuation of section 1.3 on macromolecules. Today we're going to talk about lipids, proteins, and nucleic acids. So first we have lipids. Lipids are also made of carbon, hydrogen, and oxygen, just like carbohydrates were, but it's generally not a designated ratio like carbohydrates was. They are typically fats, oils, and waxes. Their function is for energy storage. That's the one we'll talk about the most, but we will also talk about the formation of plasma membranes, and they're also used for insulation in a lot of marine animals that live in cold climates. The monomer, there really is not a true monomer to a lipid. It is made of a glycerol and any number of fatty acids linked together. So as a result of that, a lipid is not a true polymer. It is really just a large macromolecule made of a glycerol linked to some fatty acids. So this is the one macromolecule that is not made of repeating subunits or individual monomers linked together. And one significant item to note about lipids is that 1 gram of fat stores more than twice the energy in 1 gram of starch, which is a carbohydrate. So generally, lipids provide more energy than carbohydrates do to living things. The structure of a lipid, most of them are called triglycerides. So here's an example of one here, which means that we have one glycerol molecule and it's connected to 3 fatty acid chains. The connection here between them, this linkage that's formed, is called an ester bond, and that is formed from dehydration synthesis, the same way that our monomers are linked together. So we have 2 hydroxyl groups here. They lose 1 hydroxyl group and 1 hydrogen, and that combines together to form this bond between the oxygen and the remaining carbons, and that bond is called an ester bond. It is a covalent bond. They are covalently linked together to make them a giant molecule, a macromolecule. There are 2 different types of lipids. One is a saturated lipid, and the other is an unsaturated fatty acid. A saturated fatty acid is one where all of the bonds that link the carbons together are all single bonds, as you can see here in the diagram. That means the atoms are packed much closer together, and this makes it a solid at room temperature. That's generally a way for you to identify if something is a saturated or an unsaturated fatty acid. It is very hard for enzymes to break it down because of how densely compacted the molecule is. And the molecule is one big, long, kind of solid chain here. So it actually causes plaques inside the body. An example of a saturated fat that is solid at room temperature is butter. An unsaturated fat, it has some double bonds within its hydrocarbon chain, its fatty acid chain. This creates bends in the molecule. That double bond causes a little bend in that fatty acid chain, and this makes it liquid at room temperature, something like vegetable oil, or corn oil, canola oil, peanut oil, soybean oil, olive oil. They're all liquid at room temperature. They are considered to be unsaturated fats. It's actually easier for enzymes to break this molecule down, and generally they are considered to be healthier than their saturated counterpart. Some common lipids that you should be familiar with. One of them is octane. That's the hydrocarbon in gasoline. It is very similar to a fatty acid that we have inside our body. Phospholipids, you should recognize that name. This is where we talk about that makes the lipid bilayer of the cell membrane. It is comprised of a glycerol, a phosphate, which is where the phospholipid comes from, and 2 fatty acids. If you recall, the structure of the phospholipid is to have a phosphate head and 2 fatty acid tails. So the tails repel water. That's where the lipid component is with the fatty acids. And then the phosphate head attracts water, which allows it to assemble into a double layer or the lipid bilayer. Another common lipid is cholesterol. Cholesterol is actually a carbon skeleton with 4 infused rings. So it's not a chain structure, it's actually a ring structure. Cholesterol is an important part of the plasma membrane as well, and it is actually the building block of all of the steroid hormones like testosterone and estrogen. They are all actually lipids. Proteins is our next one. Proteins have nitrogen added to them in addition to the carbon, hydrogen, oxygen that we've seen in all of the others. They are found in meat, fish, dairy, nuts, and grains. They have a wide variety of functions that we will talk about at another time. The monomer is amino acids. We will definitely talk about the structures of those. And then the polymer, when you link them together, they can either be a dipeptide when you have 2 amino acids linked together, or a polypeptide when you have many amino acids linked together. These are all the different functions of proteins. You should be familiar with these. You do not have to memorize this list, but you should be able to recognize some of the things that proteins can do. More importantly, though, is the structure of the amino acid. There are 30 different amino acids, and each have a different side chain or R group. Those R groups are what determine the characteristics or the properties of that particular amino acid. If they're hydrophobic, hydrophilic, how they link to each other. All amino acids also have an amino group on one side and a carboxyl group on the other. This structure is something that you should be able to recognize, and you should be able to identify both the amino group, their carboxyl group, and the side chain that comes out from this carbon base here. These side chains contribute to the folding of the protein. We're going to talk about that as well. They interact with each other, and they interact with water, and that determines how a chain of amino acids gets folded. And again, we have dehydration synthesis that would link these together, where you have this nice hydroxyl group here, and on the next amino acid would have this hydrogen, so you pull the water out, and now you have what's formed is a peptide bond. So the bond for proteins is a peptide bond. There are different structures that we're going to talk about within a protein. First is the primary structure. The primary structure is nothing more than the linear chain of amino acids. So the individual order that the amino acids link together and line up in this chain is considered to be the primary structure of an amino acid. This is defined by the DNA code. As you know, that all of those amino acids are determined by the 3-letter DNA codes that are interpreted during protein synthesis. The bonds that are involved in this, these are those peptide bonds that we just talked about that connect between multiple amino acids. Again, it's a covalent bond. It is a strong bond. If we go back here, the bond is formed between this carbon and the nitrogen of the neighboring amino acid through dehydration synthesis. The secondary structure of a protein, there are 2 different things that can happen here. The secondary structure is where that amino acid chain is either coiled or folded. So it can be coiled up into an alpha helix, which is the structure right here, or it can be folded into what's called a beta pleated sheet, which is kind of a zigzagging sheet. If you took a piece of paper and folded it like a fan to make a fan out of it, that would be like a beta pleated sheet. This is caused by the hydrogen bonds between the amino and the carboxyl groups of individual amino acids. The oxygen of the carboxyl group forms a hydrogen bond with the HN of the amino group on the next amino acid in the chain. Remember, a hydrogen bond is just a weak interaction that holds two molecules together because of an uneven sharing of electrons. So the bonds involved with the secondary structure of a protein are hydrogen bonds. Those hydrogen bonds cause either coiling into an alpha helix or folding into a beta pleated sheet. The tertiary structure of a protein is when the whole molecule folds. So you have many different bonds that are involved here. These are caused by the interactions between those side groups, those R groups. You can have hydrophobic interactions where you have a nonpolar side chain. It's that cluster in the center of the protein away from water. Nonpolar is going to be hydrophobic. A hydrogen bond can form between some of those polar side chains where we have that uneven sharing of electrons causing hydrogen bonds. You can have ionic bonds that form between charged side chains. Some of those R groups have a positive or negative charge to them. And then if any of them have a sulfur in it, it may form what is called a disulfide. That's supposed to be bridges that can form between those particular side chains. So there are a lot of bonds involved in the tertiary structure where you have multiple folding of the whole protein. And then the last one is the quaternary structure. This is where you have multiple polypeptide chains that all fold together. An example is collagen. Collagen is a triple helix, so it's made of 3 individual polypeptides that are connected together. Hemoglobin is also an example where you have 4 subunits that are joined together and they carry oxygen in the blood. The bonds involved are the same as the tertiary structure. Sickle cell anemia is an example of a disorder that can result from a change in one amino acid. That one amino acid causes the entire chain of that protein to be shaped differently, and it causes a very, very significant issue in individuals who have sickle cell anemia. Proteins can also denature and renature. Denaturing is when proteins are misfolded because of a change in the protein's environment that affects those bonds. So all of those bonds that we just talked about, the hydrophobic interactions, the ionic bonds between the side chains, hydrogen bonds, if the proteins are put into an environment that causes those bonds to change, then it could cause the misfolding of that protein, which will cause the protein to not function anymore. Sometimes, if the environment is reversed, a protein can return to its correct folding, but not all proteins can do this. Not all proteins can renature or go back to their original form. Some causes of denaturation include the transfer to a nonpolar solvent, the change in pH of a solution, and excessive amounts of heat. Chaperonins are proteins that supervise the folding of other proteins. They actually provide a secure hydrophilic environment so that the protein, the polypeptide chain, goes into it and it gets folded up inside there safely, and then the correctly folded protein exits from the chaperonin. So this has actually been indicated in as an error, an issue in a lot of diseases like Alzheimer's and Parkinson's disease, because those are all connected to misfolded proteins. Our last macromolecule are nucleic acids. They have carbon, hydrogen, oxygen, nitrogen, and they have phosphates involved. We've talked about these quite a bit previously. DNA, RNA are the primary examples. We also have some other molecules that are involved with energy, like ATP. The monomer is a nucleotide, and the polymer is a nucleic acid. Structurally, we have a sugar, a phosphate, a nitrogen-containing base. The sugars and phosphates join together to make a backbone. Those bonds are phosphodiester bonds. And then the nitrogen-containing bases in the middle. Those nucleotides are linked together by hydrogen bonds. And that is it for the structure of the